Multiplication of TMV and bacteriophage. Introduction to bacteriophage. Bacteriophage is a virus that infects and replicates within bacteria. Bacteriophages are composed of proteins that encapsulate a DNA or RNA genome and may have relatively simple or elaborate structures. Their genome may encode as few as 4 genes and as many as hundreds of genes. Phage replicate within bacteria following the injection of their genome into the cytoplasm. Bacteriophage are among the most common and diverse entities in the biosphere. Phages are widely distributed in locations populated by bacterial hosts such as soil or the intestines of animals. One of the densest natural sources for phages and other viruses is seawater, where up to 9 into 10 power 8 virions per millimeter have been found in microbial mats at the surface, and up to 70% of marine bacteria may be infected by phages. They have been used for over 90 years as an alternative to antibiotics in the former Soviet Union and Central Europe as well as in France. They are seen as a possible therapy against multi-drug resistant strains of many bacteria. Let us now look at the replication of a bacteriophage. Bacteriophages may have a lytic cycle or a lysogenic cycle. Lysogeny or the lysogenic cycle is one of the two methods of viral reproduction. The lytic cycle is the other. Lysogeny is characterized by integration of the bacteriophage nucleic acid into the host bacterium's genome or formation of a circular replicon in the bacterium's cytoplasm. In this condition, the bacterium continues to live and reproduce normally. The genetic material of the bacteriophage called a prophage. A prophage is a bacteriophage, often shortened to phage, genome inserted and integrated into the circular bacterial DNA chromosome or existing as an extra chromosomal plasmid. There is a latent form of a phage in which the viral genes are present in the bacterium without causing disruption of the bacterial cell. It can be transmitted to daughter cells at each subsequent cell division, and later event can release it, causing proliferation of new phages via the lytic cycle. Lysogenic cycles can also occur in eukaryotes, although the method of DNA incorporation is not fully understood. The distinction between lysogenic and lytic cycles is that the spread of the viral DNA occurs through the usual prokaryotic reproduction, while the lytic phage is spread through the production of thousands of individual phages capable of surviving and infecting other cells. The key difference between the lytic cycle and the lysogenic cycle is that the lysogenic cycle does not lyse the host cell. Phages that replicate only via the lytic cycle are known as virulent phages, while phages that replicate using both lytic and lysogenic cycles are known as temperate phages. In the lysogenic cycle, the phage DNA first integrates into the bacterial chromosome to produce the prophage. When the bacterium reproduces, the prophage is also copied and is present in each of the daughter cells. The daughter cells can continue to replicate with the prophage present, or the prophage can exit the bacterial chromosome to initiate the lytic cycle. And a few viruses are capable of carrying out both. With lytic phages such as the T4 phage, bacterial cells are broken open and destroyed after immediate replication of the virion. As soon as the cell is destroyed, the phage progeny can find new hosts to infect. Lytic phages are more suitable for phage therapy. Some lytic phages undergo a phenomenon known as lysis inhibition, where completed phage progeny will not immediately lyse out of the cell if extracellular phage concentrations are high. This mechanism is not identical to that of temperate phages. In virology, temperate refers to the ability of some bacteriophages to display a lysogenic life cycle. Many temperate phages can integrate their genome into the host bacterium's chromosome, together becoming a lysogen as a phage genome becomes a prophage. A temperate phage is also able to undergo a productive, typically lytic life cycle where the prophage is expressed, replicates the phage genome and produces phage progeny, which then leave the bacterium. With phage, the term virulent is often used as an antonym to temperature. But more strictly, a virulent phage is one that has lost its ability to display lysogeny through mutation rather than a phage lineage with no genetic potential to ever display lysogenic property would be described as going dormant and is usually temporary. In contrast, the lysogenic cycle does not result in immediate lysing of the host cell. Those phages able to undergo lysogeny are known as temperate phages. Their viral genome will integrate with the host DNA and replicate along with it fairly harmlessly, or may even become established as a plasmid. The virus remains dormant until host conditions deteriorate, perhaps due to depletion of nutrients. Then the endogenous phages, endogenous substances, are those that originate from an organism, tissue or cell. Endogenous viral elements are DNA sequences derived from viruses that are ancestrally inserted into the genome of germ cells become active. At this point, they initiate the reproductive cycle, resulting in lysis of the host cell. As the lysogenic cycle allows the host cell to continue to survive and reproduce, the virus is reproduced in all of the cell's offspring. An example of a bacteriophage known to follow the lysogenic cycle and the lytic cycle is the phage lambda of E. coli. Let us see the life cycle. The lytic life cycle takes approximately 30 minutes. In this life cycle is from entering a bacterium to its destruction and usually occurs in 37 degrees centigrade. Adsorption and penetration almost starts immediately, which is followed by arrest of host gene expression, which also starts immediately. Enzyme synthesis is the next step and it takes about 5 minutes, which is then followed by DNA replication, which starts after 10 minutes. The next stage is the formation of the new virus particle, which starts after 12 minutes. After the life cycle is complete, the host cell bursts open and ejects the newly built viruses into the environment, destroying the host cell. T4 has a burst size of approximately 100 to 150 viral particles per infected host. Complementation, deletion and recombination tests can be used to map out the R2 gene locus using T4. These bacteriophage infect a host cell with their information and then blow up the host cell, thereby propagating themselves. Sometimes prophages may provide benefits to the host bacterium while they are dormant by adding new functions to the bacterial genome in a phenomenon called lysogenic conversion. Examples are the conversions of harmless strains of Corynebacterium diphtheriae or Vibrio cholera by bacteriophages to a highly virulent ones, which cause diphtheria or cholera respectively. Strategies to combat certain bacterial infections by targeting these toxin encoding prophages have been proposed. Let us now study the attachment and penetration of a bacteriophage. To enter a host cell, bacteriophages attach to specific receptors on surface of bacteria including lipopolysaccharides, teichoic acids, proteins or even the flagella. This specificity means a bacteriophage can infect only certain bacteria bearing receptors to which they can bind, which in turn determines the phage's host range. Host growth conditions also influence the ability of a phage to attach and invade them. As phage virions do not move independently, they must rely on random encounters with the right receptors when in solution, that is, when in contact with blood, lymphatic circulation, irrigation, soil, water, etc. Myovirus bacteriophages use a hypodermic syringe-like motion to inject their genetic material into the cell. After marking contact with the appropriate receptor, the tail fibers flex to bring the base plate closer to the surface of the cell. This is known as reversible binding. Once attached completely, irreversible binding is initiated and the tail contracts, possibly with the help of ATP present in the tail, injecting genetic material through the bacterial membrane. Podoviruses lack an elongated tail sheath similar to that of a myovirus. So, they instead use their small tooth-like tail fibers to enzymatically degrade a portion of the cell membrane before inserting their genetic material. How do bacteriophages synthesize proteins and nucleic acids? Within minutes, bacterial ribosomes start translating viral mRNA into protein. For RNA-based phages, RNA replicase is synthesized early in the process. Proteins modify the bacteria RNA polymerase so it preferentially transcribes viral mRNA. The host's normal synthesis of proteins and nucleic acids is disrupted and it is forced to manufacture viral products instead. These products go on to become part of the new virions within the cell, helper proteins that help assemble new virions or proteins involved in cell lysis. Walter Fires was the first to establish complete nucleotide sequence of a gene and of the viral genome of bacteriophage MS2 in 1976. Let us see how the virion assembly occurs. In the case of a T4 phage, the construction of new virus particles involves the assistance of helper proteins. The base plates are assembled first, with the tails being built upon them afterwards. The head capsids, constructed separately, will spontaneously assemble with the tails. The DNA is packed efficiently within the heads. Whole process takes about 15 minutes. The next step is release of virions. Phages may be released via cell lysis by extrusion or in a few cases by budding. Lysis by tailed phages is achieved by an enzyme called endolysin which attacks and breaks down the cell wall peptidoglycan. An altogether different phage type, the filamentous phages, make the host cell continually secrete new virus particles. Released virions are described as free and unless defective are capable of infecting a new bacterium. Budding is associated with certain mycoplasma phages. In contrast to virion release, phages displaying a lysogenic cycle do not kill the host, but rather become long-term residents as prophage. Now, Let us move into the category of tobacco mosaic virus, which is also known as TMV. TMV is a positive sense single-stranded RNA virus that infects plants, especially tobacco and other members of the family Solanaceae. The infection causes characteristic patterns such as mosaic-like mottling and discoloration on the leaves. Hence, it gets its name tobacco mosaic virus. TMV was the first virus to ever be discovered. Although it was known from the late 19th century that an infectious disease was damaging tobacco crops, it was not until 1930 that the infectious agent was determined to be a virus. Tobacco mosaic virus has a rod-like appearance. Its capsid is made from 2130 molecules of coat protein and 1 molecule of genome single-stranded RNA 6400 bases long. The coat protein self-assembles into the rod-like helical structure around the RNA, which forms a hairpin loop structure. The protein monomer consists of 158 amino acids which are assembled into 4 main alpha helices which are joined by a prominent loop proximal to the axis of the virion. Virions are negatively stained electron microphotographs show an inner distant channel of around 4 nanometers. There are 3 RNA nucleotides per protein monomer. The X-ray fiber diffraction structure of the intact virus was studied based on the electron density map 3.6 Armstrong resolution. Let us look at the disease cycle of TMV. TMV does not have a distinct overwintering structure. Rather, it will overwinter in infected tobacco stalks and leaves in the soil, on the surface of contaminated seed. TMV can even survive in contaminated tobacco products for many years. With the direct contact with host plants through its vectors, which are normally insects such as aphids and leafhoppers, TMV will go through the infection process and then the replication process. What exactly happens in the infection process? After its multiplication, it enters the neighboring cells through plasmodesmat, which are microscopic channels which traverse the cell walls of plant cells and some algal cells, enabling transport and communication between them. For its smooth entry, TMV produces a 30 kDa movement protein. Movement proteins are non-structural proteins encoded by many, if not all, plant viruses to enable their movement from one infected cell to the neighboring cell. Some plant viruses express more than one movement protein called P30 which enlarge the plasmodesmata. TMV most likely moves from cell to cell as a complex of the RNA, P30 and replicase proteins. It can also spread through phloem. Phloem is the living tissue that carries organic nutrients for longer distance movement within the plant. Moreover, TMV can be transmitted from one plant to another by direct contact. Although TMV does not have defined transmission vectors, the virus can be easily transmitted from the infected host to the healthy plants by human handling. Followed by infection, now let us see how does it replicate. Following entry into its host via mechanical inoculation, TMV uncoats itself to release its viral positive RNA strand. As uncoating occurs, the MET-HEL Pol gene is translated to make the capping enzyme MET-HEL and the RNA polymerase. Then, the viral genome will further replicate to produce multiple mRNAs via the negative RNA intermediate primed by the tRNAs at the positive RNA third end. The resulting mRNAs encode several proteins including the coat protein and the RNA-dependent RNA polymerase, as well as the movement proteins. Thus, TMV can replicate its own genome. After the coat of protein and RNA genome of TMV have been synthesized, they spontaneously assemble into complete TMV virions in a highly organized process. The protomers come together to form discs or lock washers composed of two layers of protomers arranged in a helix. The helical capsid grows by the addition of protomers to the end of the rod. As the rod lengthens, the RNA passes through a channel in its center and forms a loop at the growing end. In this way, the RNA can easily fit as a spiral into the interior of the helical capsid. This is the life cycle of a TMV virus. Let us look at the host and the symptoms. Like other plant pathogenic viruses, TMV has a wide variety of host range and has different effects depending on the host being infected. The tobacco mosaic virus has been known to cause production loss for flue cured tobacco of up to 2% in North Carolina. It is known to infect members of 9 plant families and at least 125 individual species including tobacco, tomato, pepper, cucumbers and a number of ornamental flowers. There are many different strains. The first symptom of this virus disease is a light green coloration between the veins of young leaves. This is followed quickly by the development of mosaic or mottled pattern of light and dark green areas on the leaves. Rugosity may also be seen where the infected plant leaves display small localized random wrinkles. These symptoms develop quickly and are more pronounced on younger leaves. Mosaic does not result in plant death, but if the infection occurs early in the season, plants are stunted. Lower leaves are subjected to mosaic burn, especially during periods of hot and dry weather. In these cases, large dead areas develop in the leaves. This constitutes one of the most destructive phases of tobacco mosaic virus infection. Infected leaves may be crinkled, puckered or elongated. However, if TMV infects crops like grapes and apple, it is almost symptomless. The environment in which TMV thrives. TMV is known as one of the most stable viruses. It has a very wide survival range. As long as the surrounding temperature remains below approximately 40 degrees Celsius, TMV can sustain its stable form. All it needs is a host to infect. If necessary, greenhouses and botanical gardens would provide the most favorable condition for TMV to spread out due to the high population density of the possible hosts and the constant temperature throughout the year. Finally, how do you follow the treatment and management practices? One of the common control methods of TMV is sanitation, which includes removing infected plants and washing hands in between each planting. Crop rotation should also be employed to avoid infected soil or seed beds for at least 2 years. As far as any plant disease, looking for resistant strains against TMV may also be advised. Furthermore, the cross-production method can be administered where the stronger strain of TMV infection is inhibited by infecting the host plant with a mild strain of TMV similar to the effect of vaccine. In the past 10 years, the application of genetic engineering on a host plant genome has been developed to allow the host plant to produce the TMV coat protein within their cells. It was hypothesized that the TMV genome will be recoated rapidly upon entering the host cell. Thus, it prevents the initiation of TMV replication. Later, it was found that the mechanism was that protects the host from viral genome insertion is through gene slicing. Tobacco mosaic virus survives on infected seeds, plant debris and even clothing for months or years. The virus enters plants through wounds sustained in transplanting or pruning. It spreads rapidly once it is in the host. The virus can remain viable for years on a dried plant debris and is extremely tolerant of very high temperatures. Tobacco products are the most common source of inoculum. Smokers can infect plants by handling them. The virus is not spread by aphids. Thus, I conclude by saying TMV can cause a deadly disease in tobacco plants. Thank you.